You know, inside every single one of us, there's this non-stop factory that's constantly at work creating every single blood cell we have. It's an incredible process, and it's called hematopoiesis. So, in this explainer, we're going to break down exactly how this factory operates, from its very first spark in the embryo all the way to the complex control systems that keep us alive and healthy every single day. All right, so here's our game plan. We'll kick things off with the basics, what hematopoiesis even is. Then we'll get into the blueprint, how it's all regulated. From there, we'll jump in a time machine and go back to its origin story in the embryo and see how it all changes after birth. We'll wrap things up by looking at the body's inventory system for these cells, and finally, how we measure the factory's total output.
Okay, let's dive right in. First things first, what exactly is hematopoiesis? Well, at its core, it's the whole dynamic process of making and developing all of our blood cells. And what's truly amazing is the powerful evidence showing that every single one of these cells, we're talking red cells, white cells, platelets, all of them, they all come from one single common ancestor, the hematopoietic stem cell, or HSC for short. This whole system is in a constant state of turnover, a relentless cycle of production to keep our blood cell populations right where they need to be. And this constant production is absolutely crucial because each cell type has a vital job to do. Think of erythrocytes, our red blood cells, like a massive fleet of delivery trucks shuttling oxygen around and hauling away carbon dioxide. Our granulocytes? They're the soldiers on the front lines fighting off infection. Lymphocytes act as our body's intelligence agency, supporting both cellular and humoral immunity. And platelets? Well, they're the emergency repair crew in charge of hemostasis. That's the process that stops us from bleeding. Now you can imagine, a factory this complex and this important can't just run on its own. It needs a very sophisticated management system, and that brings us to regulation and control, the blueprint that governs the whole operation. Probably the best way to picture this is as a hierarchy, kind of like a family tree. Right at the very top, you have the hematopoietic stem cells. They give rise to precursor cells that start committing to specific career paths, deciding they're going to become a red cell, or a white cell, or a platelet. This entire pyramid of development is supported and directed by these little chemical messengers called cytokines.
And when we talk about cytokines, we're talking about a really diverse group of key players. They include families of molecules like interleukins, or ILs, and colony-stimulating factors, CSFs, as well as interferons. And you probably recognize a couple of the really famous ones, like erythropoietin, or EPO, which is the signal to make more red blood cells, and thrombopoietin, TPO, which does the same thing for platelets.
So how does a stem cell actually decide what it wants to be when it grows up? Well, that process is called differentiation. As a cell moves down that hierarchy we just talked about, it loses some of its ability to self-renew and becomes more and more committed to one specific path. This whole decision-making process is controlled by a network of transcription factors. You can think of these like tiny molecular switches that find specific DNA sequences and flip genes on or off. That's what guides the unique gene expression that makes a red blood cell different from a lymphocyte.
So most of this action happens in the bone marrow, but these super important stem cells aren't just floating around randomly. No, they live in these specialized microenvironments called niches. The stem cell niche is this highly regulated dynamic structure. It provides all the signals needed to support the core properties of HSCs, their ability to make more of themselves, and to repopulate all the different blood lineages for our entire lives. It's constantly modulating their growth and differentiation based on what the body needs. And we're now learning that immune cells, like macrophages, are also critical regulators of this whole environment.
And what's really wild is just how many different cell types are part of this support team. I mean, look at this. You have endothelial cells and mesenchymal stromal cells. Those are the major regulators, absolutely required to maintain that stem cell pool. But then you also have neural Schwann cells, megakaryocytes, macrophages, and even osteoblasts. They all have these key roles influencing the stem cell cycle, where they're located, and what they ultimately become.
Okay, so we've seen how it works, but where did it all begin? Every great system has an origin story, and for hematopoiesis, that story starts way, way before we're even born.
So in the first few weeks of embryonic life, hematopoiesis actually kicks off in the yolk sac. This early production starts to decline at about 6 weeks, and pretty much stops by the 2-month mark. At that point, the fetal liver takes over as the main production site, and it holds that job until the 7th month. Now, from the 3rd to the 6th month, the spleen also pitches in. And then finally, around the 7th month of fetal life, the responsibility shifts to the bone marrow, which becomes the primary site for blood cell development from then on. And as the bone marrow starts to come online, you see it just get packed with red blood cells. The bones in the toes, fingers, vertebrae, and cranium are all just filled with these erythroid cells. We think early lymphocytic cells might also form during fetal life. We see the first megakaryocytes, those are the platelet precursors, show up at about 3 months, and then granulocytes are observed around the 5-month mark.
All right, that's the story before birth. But what happens once we enter the world? Well, the location and the intensity of this cellular factory keep changing throughout our lives.
So, after we're born, the liver and spleen stop their production, and our bone cavities, the red marrow, take over completely. In infancy, this is happening across the entire medullary space of all our bones. But a change starts happening after age 4. That's when fat cells which create yellow marrow start appearing in the long bones. This process of consolidation continues, so that by the time you're about 18 to 20 years old, active marrow is really only found in specific places: the sternum, ribs, pelvis, vertebrae, and skull. Then, later in life, after age 40, the active marrow in those spots is made up of about 50% hemopoietic tissue and 50% fat.
But our bodies are smart, right? What if we need a major surge in production because of an infection or some kind of bleeding? Well, the system has a backup plan. When there's a huge demand for blood cells, active hematopoiesis can actually fire back up in the spleen, liver, and other tissues. This amazing compensatory mechanism is known as extramedullaryhematopoiesis. Okay, let's shift gears and talk about inventory management. The body has this really clever way of maintaining different pools of cells. Some are out in the field, doing their jobs, while others are kept in storage, ready to be deployed at a moment's notice. Let's compare the inventory for a couple of different cell types. For granulocytes, the cells out in our peripheral blood are split pretty much 50-50. Half of them are circulating and functional, doing their thing. The other half are in storage, basically lining the walls of our blood vessels in what we call the marginating pool. Platelets, though, are managed a little differently. About 70% of them are circulating in the blood, while the other 30% are stored away in the spleen, just waiting for the call to action. But then you have the red blood cells, the erythrocytes, and they're a completely different story. There's no storage pool out in the periphery for them. 100% of mature red blood cells are circulating in the peripheral blood in a functional state, constantly working.
All right, last section. We've talked about the factory and the inventory. Now, how do we actually check the factory's productivity? That brings us to our final topic, measuring factory output using bone marrow cellularity and some key ratios.
We can actually get a pretty good estimate of how active someone's marrow should be. At birth, normal cellularity is 100%. In adults, it settles down to about 50%. There's a neat little rule of thumb to estimate the normal range for any age. Just take 100, subtract the person's age, and then it's plus or minus 10. So, for example, if you have a 50-year-old person, their normal marrow cellularity would be in the range of 40% to 60%.
And within that active marrow, there's a very specific balance we look for. The average myeloidtoerythroidratio, or MTE ratio, is about 4:1. Remember, myeloid cells are the precursors for granulocytes, and erythroid cells are the precursors for red blood cells. So if we take our typical adult with 50% marrow cellularity, about 40% of that active tissue is dedicated to making granulocytes, that's granulopoiesis, and about 10% is dedicated to making red cells, which is erythropoiesis.